Okay, today I'm going to talk about using a piece of apparatus called a hemocytometer. It's a specialised slide. It's quite solid. It's basically a precision piece of glassware. I don't know if you can see, there's a silvered surface here. It's like an H, but actually in there and in there's a cross piece. Now I'll show you what that has on it. If you look at the board, okay, that's the slide there. If you look at the diagram, there's the cross pieces here and here, and then if you put a cover slip, there's a thin gap there. Now if you look at that in detail, that cross piece is precision cut, laser cut lines. So you've got big squares, rectangles, and then in the middle is small squares. And it's actually these small squares we're going to be using to count cells. These are the small squares here. So when you look at it, you need to focus on the small squares. So if you come back to this, what you will do, you'll place your slide in the slide holder. It's clipped in. You'll move the slide. I don't know if you can see the middle of one of these two rectangular areas under your lowest magnification, the red lens, which is a 4x lens. And then focus away until you see the small squares. On the lowest magnification, centre those small squares right in the middle of what you're looking at. And then you need to focus on the next lens. Again, focus until you see the small squares. Move them, and so the small squares in the middle. And then again on the 40x the blue lens. I'm actually going to use the blue lens for counting cells. Now unfortunately you can't show you what I'm looking at, but I've got a picture taken by a student using their mobile phone, which is quite a handy technique. And so, I don't know if you can see, the squares are actually in blocks of 16. Now she's put some yeast cells on it and then covered it with a cover slip. And she was able to count each of these as a cell. So this square, there's 5 cells, this square is 1, this square there's none, this square there's 1 there, 3 there, 1, 2, 4 there, none there. Now let's have a look at how we apply cells to that. So if I just move that to one side, I'm not going to unfocus it because once it's adjusted it should be nearly in focus. These are quite expensive, so please be careful with them. And so here's our yeast culture, which you're going to check how many cells per mil there are in this culture. It's growing in 10% sucrose, and we know it's growing because it's bubbling, producing bubbles of carbon dioxide. Now if I put some of this directly on that slide, I know there's too many cells, so we actually need to make a dilution. So in fact what you'll do is when you'll put some yeast in a test tube. You don't need a lot. I've already labelled this marks yeast culture. This will go in an incubator, and then you will every 15 minutes you'll take a sample of that. Now I'm going to make a 1 in 10 dilution, just like you did with the bacteria. If you notice, I'm not worried about being sterile. I've got some tap water here. So I've got this tube here labelled 0. This is because it's my first reading. Okay, I'm going to put 900 microliters of water. I'm not worried about being sterile. In fact, you don't need to wear lab coats for this experiment. I'm going to take 100 microliters of this culture, and I'm going to put it into there. So it's made it much more dilute. As soon as I've done that, I'm going to put my growing yeast into the water bath, and I'm going to start the clock. So in 15 minutes time I'm going to take another sample out of there and dilute it. In fact, I've got my dilution tubes ready. I've got one labelled 15, already with water in it, 30, 45, and if we're lucky, 60 minutes. So I'm going to do samples every 15 minutes. So now I've got my first sample ready. I'm going to take a fresh yellow tip. It's always worth mixing this just to make sure. See how I use a little finger just to give it a little tap. Take up 100 microliters. The precise volume is not too important for this bit. I am going to put that on there so you can see. Put the cells on there. It doesn't really matter where you put it. Now, I'm going to put the cover slip over there. It squashes the solution over the slide. You won't see anything. And then I'm going to focus that. Unfortunately, I can't show you what I'm seeing, but you'll see something like that. What is on the screen. So focusing that. Okay, this is a bit of a skill, so the first few times you must make sure you put the small squares. And I've got nice cells there. I can even see some dividing. Now, what you then have to do is count these cells. So you actually need to do some maths. Okay? Because you've got to work out cells per mil is the dilution, Bunington factor, times the number of cells you count, times 4,000, divided by the number of squares counted. Now the easiest thing to do, actually, is to focus on a block of 16. 16 squares. Count all the cells. 5, 6, 0, 0, 7, 8, 9, 10, 11, 12, 13, 14. And so I count a whole block. So we caught, say, 50 cells. It's in 16 squares. So use the formula. 10 times, whatever it was, 53 cells, times 4,000, divided by the number of squares, often 16. You don't have to count 16 squares, but it's often the easiest thing to do. Do the maths and you'll get it something between 10 to 100,000 cells per mil. You then need to clean your slide off. So you just take this off carefully, rinse that. You can dry that, and you can rinse off the cover slip. The cover slip, by the way, is actually the main health and safety issue here. So there's a good chance you're going to break this. I will issue with just one of these. If you break this, it must go into the glass disposal bin. Okay? Do not put this on the bench because they disappear. But the actual issue is, if there's any of these broken on the bench or in the sink, they're actually a hazard for the next class and a hazard for the cleaners. So I won't let you get another one until you've told me that you've actually disposed of it correctly in the glass bin, not the normal waste bin. And then in 15 minutes time, you can go over, get your 15-minute tube ready, fresh tip, take your tube, give it a quick tap, because they do tend to settle out. Keep the timer running, don't stop the timer. Take out your 15-minute sample, put that back in the water bath, and then go and put this back in your dilution tube. So this is now my 15-minute reading. Again, put this onto the hemocytometer, take your reading. Hopefully we'll see a small increase in the number of cells. And then you'll do the same thing at 30 minutes, 45 minutes, and if it goes well, 60 minutes. And then we'll plot the results on a line graph.